Hello everyone. Today we discuss the workstation of the IVF lab. So we discuss the how to work and how to regulate this equipment. So we discuss the first of the HEPA filter. In here, put the HEPA filter. They purify the air like bacteria and any pollen grains and any type of present in the air. So we eliminate the all types of bacteria and viruses and also fungi. So we give the sterile environment of the embryo and sperm. So this is the pre-processing sample in here we done. So first we discuss the HEPA filter. In here put the vertical HEPA filter. So in here air flow the vertical and surface by the clean. And two types of HEPA filter, vertical and horizontal, but in here the vertical. So when we any process done, we start the air in here, this and this. This is the increase the airflow and this is the decrease the airflow. So this HEPA filter 99.97% purified. So this is the warmer. In here we put the sample like semen and embryo and done the any procedure. This is the special type of LED light. This is the LS1112. This is like the all weather condition working. So in here light we increase and decrease. In here decrease light. In here increase the light. So this is the increase and decrease the light. And this is the heat plate. This is 37 Celsius, maintain the temperature. Because of the all our cells, sperms and embryo, they work the 37 Celsius. Why 37 Celsius degree required for sperm and embryo? Because of 37 is the optimum temperature of the cells. In 37 degree, cell divide the properly and working the DNA and ion exchange properly, and enzyme also very work in a 37 degree. So we maintain the 37 Celsius and this is the thermometer. We increase and decrease the temperature in here. This machine is automatic regulated, so we do not decrease and increase the 37 Celsius. So this is the warmer. And this is the CO2 incubator. This is the maintain the optimum pH in here and the optimum pH in here. So in here we use CO2, not 100% CO2. We use a concentration CO2. So this is the maintain the optimum pH. So and here this is the light. So we off on light because of the when we working the and see the proper of the cells. And this is the UV light, but UV light not inject in here because UV light we do not use in IVF and ICSI lab. So this is the warmer and this is the lights. So we discussed the what is the mechanism of working of the HEPA filters. So first we impaction. So in the impaction we trap the large particle in the air. So this is the impaction. Air flow in this direction. So this is the fibers. Last one is the diffusion. So in the diffusion, the size of particle is like gaseous form. So this is very small and little size of particles in a diffusion. So they do not flow the in direction of the air. They flow the Brownian motion. So this type of motion, this. So they struck in here. So this is the diffusion. They trap in here. So this is the gaseous particles. So we we discuss the third type of impaction, interception, and diffusion. Impaction is a large size of molecules. Interception is a medium size of. Diffusion is a gaseous form of. So they trap in here, and impaction trap in the outside, and interception they trap in here. So what is this? This is the HEPA filter. This is the made by the polybromine and fiberglass fibers and borosilicate glass fibers. This is the polybromine is a high thermal capacity. So these HEPA filter made by the polypropylene, polypropylene. So this is the very high melting point, 171 Celsius. So this is a very good. And in this fibers, this type of, they arrange this type of, this very compact form of. So in here trap the any particles. Also in capacity, they absorb the UV absorbing power. In polyethylene also UV absorbing power. They made also in here the ferrosilicate, also mixed with ferrosilicate. So ferrosilicate is a melting point is a 150 Celsius. This boron silicate is a non-toxic. Does not absorb stains or odors. It's made from safe chemicals. It does not absorb stains or odors. So this is the reason we do not use perfume in our lab, because this ferrosilicate do not capture the odors, so they pass and cross the HEPA filter and they affect the embryo and cells and increase the VOC. So they inhibit the growth of embryo. So we do not use the perfume in a IVF lab. So that's the reason HEPA filters cannot capture the gaseous pollutants like chemicals, fumes, gases and odors. So we avoid this type of things. We do not use perfumes. So we discuss the advantages of HEPA filters. HEPA filters trap the pollens, dirt, dust and moisture and bacteria and virus and some microbes and liquid aerosol. HEPA filters cannot capture the gaseous pollutants like chemicals, fumes and gases and odors we already discussed. So advantages: improving air quality, and balance air flow. And air pressure. This is also maintain the air pressure. So we maintain the embryo and mimic the environment like a uterus. So balance the air flow. Balance air flow, they regulate the cell division, and air pressure, they also regulate the cell division. If we do not maintain the air pressure and balance airflow, they inhibit the cell division. So in a HEPA filter, we reduce the VOC compounds. So what is the VOC compound? They are the invisible and do not settle as dust as particle do. Formaldehyde, isopropanol, hydrocarbons such as xylene and refrigerant components. So this is the VOC compound. They inhibit the growth of the embryo. So we give the sterile environment or in the IVF and workstation of which we process the pre-process sampling. So this system is the work based on the mechanical. So also in a electrostatical charge based of the HEPA filters. They trap the negative and positive air dust and they trap in air. Electrostatical charge, this increase the efficiency of the trapping particles positive and negatively, like this is the negative particles air dust and this is positive columns. Okay, so in the trap in here, question arises from why we use HEPA filter in the IVF lab. Because of infertility is a common disease which drives several couples to seek treatments. So with assisted reproduction, 15% of couples around the world infertile. 15% couples is the infertile. Only we treated 85% of them can be treated with the assisted reproductive technology. So we use the HEPA filters in the infertility in the IVF lab. Let's discuss the humidifier. So humidifier injected in the workstation of the IVF lab. So the nozzle for the output gas is the fitted on the back wall of the work area next to the humidifier glass bottle. So the glass bottle is placed in the aluminium block which is heated by the table plate heating system. Maintain the pressure of workstation 7.25 psi, which regulate the also pressure. So the humidifier mixed with 5% sterile water, distilled water and 6% CO2 gas concentration and maintain the air temperature and air humidity, and reliable incubator humidifier must be secure the consistent of maintaining the proper humidity levels for it provide optimal environment for the culture medium, resulting in the successful embryo development. So and this humidifier changing the water bottle every month because of maintain the sterility, and we change the weekly. So question arises, why is CO2 essential in the humidifier? Because CO2 regulate the pH. Because in essential embryogenesis we do not use oxygen. Oxygen disturbs the pH and carbon dioxide is essential for regulating the pH of the culture media on its acts in the buffer to maintain the pH within the physiological range for the cells of the grow. This CO2 converted by the pyruvate, and in early embryogenesis, embryo give by the pyruvate, not a oxygen and glue. So pyruvate is the essential for early embryogenesis, and also pyruvate is the species specific. In a pig, in case of pig embryo, do not require the pyruvate. So this pyruvate also species specific. So this is the reason for we use CO2 in a early embryogenesis, not a O2, because of pyruvate regulate the pH and cell growth and cell division and give a proper amount of energy and regulate the metabolism. So this is the function of humidifier. So question arises, why air quality essential for in the IVF lab, because in some study found air quality is a very important for IVF and excellence, because improve fertilization rate and increase the number of blastocyst growth and increase the embryo implantation and reduce the miscarriage. So we maintain the air quality in the lab by the HEPA filters and some other instruments.